Thank you again for joining us, and thank you for submitting to Pestola. I have to say, I had to message Dr. Trigestad to find out your background because you didn't put anything on your bio. So I'm quite impressed. You have quite a diverse background. I understand that you are double major in both biology and ballet. Is that correct?
Yes, that's correct.
Oh, thank you for your time, devotion, in science. So Dr. Trigestad is, I think, very privileged to have you work in her lab. And so we're really looking forward to finishing off with some basic science and talking to us about microRNA increasing adipose signaling and decreasing proliferation of mesenchymal stem cells. Thank you.
Thank you very much. Again, my name is Olivia Brooks, and I work at the OU Children's Hospital in Oklahoma City, Oklahoma, with the CMRI Metabolic Research Program. And I'm going to be talking today about some potential epigenetic factors, specifically microRNA 126, that may potentially program infants born to mothers with diabetes to develop the disease themselves later in life. So, as a brief reminder and for a little bit of background, incidence of obesity and type 2 diabetes has increased rapidly in youth within the last 20 years. And obesity is commonly accompanied by comorbidities such as hypertension, dyslipidemia, and notably type 2 diabetes. Some recent studies have indicated that children born to mothers with diabetes are at a higher risk of developing the disease themselves later in life, as opposed to their peers who are not exposed to diabetes in utero. So what we see from this trend is that we start to see a cycle of diabetes, whereby mothers with diabetes give birth to infants who are more likely to develop the disease later. And when they become mothers themselves, they also give birth to infants who are also more likely to develop the disease. And then the cycle goes on and on. So we know that some of the cycle is attributable to genetic factors, but we also believe that there are some epigenetic factors at play here, and that's what we are interested in. Some potential epigenetic factors that our lab specifically looks at are microRNAs. So microRNAs are short non-coding pieces of RNA that are about 18 to 25 nucleotides in length, and they function to regulate gene expression at the post-transcriptional level. So this happens when a piece of mature microRNA and RISC proteins bind to their target piece of messenger RNA. And gene expression regulation can happen in two different ways from here. When there's perfect complementation between the microRNA and the messenger RNA is degraded and can no longer be translated into its proteins. If, however, there is imperfect complementation between the microRNA and messenger RNA, the translation of messenger RNA into its proteins is suppressed. So this shows that microRNA plays a big role in regulating gene expression. And when there are different levels of microRNA present, this can also impact the proteins that are available to be translated and the genes that are therefore expressed. So some previous research that our lab has also done has shown that human umbilical vein endothelial cells, or HUVEC, which have been exposed to diabetes, show differing levels of different microRNAs than do HUVEC that have not been exposed to diabetes. So this previous study from this graph over here shows that HUVEC exposed to diabetes have much higher levels of microRNA 126 than do HUVEC that have not been exposed to diabetes. And over here from this study, we can see that microRNA 126 is one of the most abundant microRNAs in HUVEC exposed to diabetes that we have assessed. Another thing that we know about microRNAs is that they can be packaged into exosomes and secreted from cells such as HUVEC. When this happens, they are put into circulation throughout the body, where they can therefore, from then, be taken up by different types of cells. And this impacts the cells that take them up. So if exposure to the diabetic milieu equals an increased level of microRNA 126 in HUVEC cells, we can assume that that also, previous studies have also shown that that increases levels of microRNA 126 in circulation, which means that they are more able to be taken up by other types of cells, such as mesenchymal stem cells. And that brings us to our current research question. So what we are currently investigating is the specific impact that microRNA 126 has on the cellular metabolism of mesenchymal stem cells, or MSCs, when MSCs take up 126 from circulation. We hypothesize that the impacts on cellular metabolism include decreasing proliferation, increasing cell cycle arrest, and increasing the capacity of MSCs to preferentially differentiate into adipocytes as opposed to other lineages. To test these hypotheses, we harvested MSCs from the umbilical veins of mothers with diabetes, which I'll refer to as our DM cells, and from the umbilical veins of mothers without diabetes, which I'll refer to as our control. The first pathway that we are looking at in these experiments is the Wnt signaling pathway, which is significant in adipogenesis. So previous studies have shown that increased exposure to microRNA 126 has the impact of downregulating its target protein, LRP6. It also downregulates the downstream protein, GSK3 beta. So when LRP6 and GSK3 beta protein are downregulated, it has the impact of blocking beta-catenin from entering the nucleus of the cell. When all of this happens, this drives the MSC further towards differentiating into an adipocyte rather than other lineages. And adipocytes are characterized by increased lipid levels and increased insulin resistance. And over here, CD13 is a cell surface marker that is associated with adipogenesis. So the higher propensity of an MSC to differentiate into an adipocyte, the more cell surface CD13 markers we would expect to see. So the first thing that we wanted to look at within this signaling pathway is how exposure to microRNA 126 actually impacts levels of LRP6 and GSK3 beta protein. Again, we would expect to see that these protein levels are decreased. So how we tested this was we transfected both our control and our DM MSCs with a non-template control mimic, a microRNA 126 mimic, and an anti-microRNA 126 mimic. After 48 hours of transfection, we then serum starved the cells for 4 hours and then stimulated them with 10 nanograms per milliliter of insulin growth factor 1. We then lysed the cells and analyzed them via Western blot. And what we found was that following transfection with microRNA 126 mimic, levels of LRP6 protein were decreased by 45% in our control cells and by 62% in our DM cells, both as compared with our non-template control transfections. Additionally, downstream, in our GSK3 beta protein, following transfection with microRNA 126, we saw a decrease by 47% in our control cells and by 24% in our DM cells. So this does indicate that it supports our hypothesis that exposure to microRNA 126 does decrease levels of LRP6 and GSK3 beta protein, which would further drive the MSC towards adipogenesis. The next pathway that we are concerned with in our experiments is how microRNA 126 impacts levels of its target proteins IRS1 and P13 kinase and phospho-AKT. Some more previous studies have shown that increased exposure to microRNA 126 decreases levels of IRS1 and P13 kinase protein and has the effect of also downregulating its downstream protein phospho-AKT. When all three of these proteins are downregulated, it has the impact of increasing likelihood of apoptosis and of increasing cell cycle arrest within MSCs as well. So the first thing that we wanted to do was look at how exposure to microRNA 126 impacts proliferation of MSCs. We did this by transfecting our control MSCs with either a non-template control mimic, our 126 mimic, or again, our anti-126 mimic. After 48 hours of transfection, we serum starved the cells overnight, and then we stimulated them with 10 nanograms per milliliter of insulin growth factor 1 for 24 hours. We then assessed proliferation levels via MTT assay. And what we saw was that following transfection with 126 mimic, proliferation levels of our control MSCs decreased by 26%, as compared with our non-template control proliferation levels. We also saw that following transfection with our anti-126 mimic, the levels of proliferation increased by 40% as compared with our our 126 transfection. So these results also support our hypothesis that exposure to microRNA 126 may impact messenger RNA targets, which have the effect of decreasing proliferation in MSCs. We also then wanted to look at how exposure to the diabetic milieu impacts cell cycle in MSCs. We did this by staining our DM and our control cells with rubidium iodine and then analyzing them via flow cytometry for cell cycle. What we found here was that there was 6% increase in the amount of DM cells found in the first G1 phase of cell cycle as compared with the amount of control cells found in this phase. And then in the next phase, S phase, we found a 38% decrease in the amount of DM cells as compared with the amount of control cells found in this phase. These results would indicate that MSCs exposed to the diabetic milieu, so our DM cells, are more likely to undergo cell cycle arrest in the G1 phase, which prevents them from proliferating and progressing into the S phase. And this has the overall impact of DM exposure leading to increased cell cycle arrest. The last thing that we wanted to look at within this signaling pathway is how exposure to microRNA 126 impacts levels of P13 kinase, IRS1, and downstream phospho-AKT within MSCs that have been differentiated into adipocytes. So how we did this was we transfected our MSCs again with our non-template control mimic, our microRNA 126 mimic, our anti-126 mimic, or leaving them untransfected. After 48 hours, we exposed the cells to 3 rounds of differentiation. We then serum starved the cells for 4 hours and then stimulated them for 10 minutes with 10 nanograms per milliliter of insulin. We then lysed the cells and analyzed them via Western blot. And what we found was that following our 126 mimic transfection, P13 kinase protein was decreased by 53% as compared with our non-template control. And IRS1 protein was decreased by 48% as compared with our non-template control. We also found in both of these proteins that exposure to anti-126 mimic preserved the P13 kinase protein expression by 42% as compared with our 126 mimic, and by 23% in our IRS1 protein as compared with our 126 mimic. You can see that in some of these blot images over here. In our downstream protein, phospho-AKT, we also saw that exposure to microRNA 126 mimic decreased protein expression by 54% as compared to our non-template control mimic. However, we did not see that same level of preservation with our anti-126 mimic in our phospho-AKT. These results, again, show that exposure to microRNA 126 does have the impact of decreasing those 3 levels of protein expression in this pathway, which would also further drive the MSC towards adipogenesis. So all of these results indicate that exposure to microRNA 126 and to the diabetic milieu does affect signaling pathways and cellular metabolism of MSCs by decreasing proliferation levels, increasing cell cycle arrest, and increasing the capacity of MSCs to differentiate into adipocytes. All of these cellular metabolism changes may potentially program infants who are exposed to diabetes in utero to develop the disease later in life. Some of our future research hopes to explore potentially new targets of microRNA 126, and specifically how microRNA 126 impacts adipogenesis. I would like to thank the NIH for their grant support with these projects, all of our collaborators and mentors in the metabolic research program, our lab staff, and I would specifically like to thank my mentor, Dr. Jeannie Trugestad, for all of her help and support with this project. And thank you very much.
Thank you. Very nice way to finish off with a strong basic science. And in your last slide, you were alluding to some of these things. So this is intriguing. So essentially, you know, I think a lot of us understand, you know, or understand epigenetics and understand that, you know, there's effects maternally, that are related to things like obesity and gestational diabetes. I'm just curious, you say in your future research and that you're looking at novel targets. What types of targets, or what specifically can you see that as far as the future direction for this research?
Yes, so I believe some of the novel targets that we're looking at are messenger RNA targets, and just also other ways that exposure to microRNA 126 may impact how the MSC differentiates into adipocytes.
Okay. We have a question as well from Dr. Triggerstadt. Can you see that on chat?
Yes. So the question says, in the experiments examining the impacts of microRNA 126 on LRP6 abundance, it appears that the LRP6 is lower in the DM cells. Do you have any thoughts on this difference? So yes, some thoughts are that in addition to exposure to microRNA 126, on the abundance of LRP6 protein, there are also some differences in just how in MSCs that have been exposed to diabetes versus MSCs that have not been exposed to diabetes. So there may be additional impacts in addition to microRNA 126 associated with diabetes that also reduce levels of LRP6 protein. And that might be something that we also further explore in our research.
Okay, thank you. Are there any other questions from the audience, either virtual or live? Well, thank you again. We're very excited, especially having someone that's, doing basic research in undergraduate. So we look forward to seeing how your career develops, and you certainly have a very good mentor in Dr. Triggerstadt. Before I sign off here for the virtual audience, we'll reconvene tomorrow at 8:30. Look forward to seeing you all. You have the lounge either on your desk, on your laptop, or on a mobile device. So jump on the lounge and do a little bit of socializing if you'd like this. And you all have a good night. And thank you again, Olivia.
Thank you very much.